Today we're going to talk about genetic management in captive populations. So why might one start a captive breeding program? Well, for several reasons. Captive breeding programs can help those incapable of surviving in their natural habitat. They are insurance for wild populations, to maintaining biodiversity by maintaining individuals in a captivity. It also can help maintain genetic health by monitoring individuals for inbreeding depression and genetic diversity. And you can then use those individuals for release back into the wild. So how specifically do the captive breeding programs themselves assist conservation as a whole? Well, one of the really useful things about these kind of programs is that they are really good at educating the public. So you can bring the public in and teach them about these species, about why to protect them, see how it works, and help fundraise for these types of conservation concerns. And a really popular fundraising technique that you probably all have heard of is the symbolic adoption of different species, where you give money and then you get flyers telling you you've adopted an animal. I know I did this when I was a kid. I think I got the wolf. It was very exciting. And I think World Wildlife Fund does this. Also, captive breeding programs are really important for basic research. A lot of times these species, we don't know a lot about them because they're rare in the wild. And when you have captive breeding programs, you can try to figure out basic research questions like, what kind of food preferences do these species have? You know, how do they live? What are their behaviors? What are their mating behaviors? And that can be really useful and important for managing them once they go back out into the wild. And then with captive breeding programs, you have a base of animals that can be used for reintroduction. So that's quite useful. So when captive breeding does occur, you're generally taking individuals from a wild environment where they have native habitats into an environment where there's captive. And they have very dramatic change in environmental features and selection pressures that are acting on them. And this can be a problem because ultimately what can happen is that species in captivity can adapt to living in captivity. So when we talk about adaptation to captivity, this is because there is a difference in the selection pressure once these individuals are brought into captivity. Captive populations are inadvertently selected for their ability to court, mate, breed, and develop in the captive environment. So there's certain traits that are actually selected against once you get into captivity. So for example, flightiness. Flightiness in animals such as antelopes, gazelles, and kangaroos. This can be selected against when in captivity because they can injure themselves by running into walls and fences and even die. Another example is tameness. So tameness is actively favored by zookeepers who want to be able to interact with the individuals. So here we have zookeepers taking a dog and a cheetah on a walk, obviously selecting for tameness there. Prey capture and predator avoidance are no longer positively selected for. There's no real need to be worried about predators or be thinking about prey and how to catch them. Now this is a really interesting picture here. This is actually taken from the National Zoo, and I've seen this before when I've gone there, and I've always thought this is really fascinating. They actually house the zebras next to the cheetahs, and so you can see that the cheetah is actually practicing, predator behavior. It's stalking the zebras. It can walk back and forth and it can follow them. So maybe that's good for the cheetahs and it mimics a wild environment. But for the zebras, they do not look very bothered by the cheetah right there, and I think that they've gained the trust and the knowledge that the cheetah is not actually going to come after them. So they would not be great candidates for reintroducing back into the wild because they've lost that fear of the cheetah. And then also veterinary care and hygiene. Because of this veterinary care and hygiene, it removes the pressure for fighting disease and parasite resistance. So these species can then become susceptible to that when they get reintroduced back into the wild. So all of these examples for adapting to captivity is detrimental when species are returned to the wild, and they end up having poor fitness consequences because of that. When we think about ways that we can minimize the genetic adaptation to captivity, this equation comes in handy because it tells us the relationship between these factors and the genetic adaptation to captivity. So this equation, let's go through it right here, says that the genetic adaptation to captivity over generations is related to S, and this is the selection pressure in captivity. This can be high or low. H2, this is the genetic variation within populations, and that's multiplied by the sum of relationship here that is 1 over 2NE. And remember, the NE is the effective population size in a captivity, and T over T minus 1 generations, and so that indicates that generations in captivity is also relevant to genetic adaptation to captivity. So again, adaptation relies on selective variability in genetic variation, effective population size, and generations. And the most effective means for minimizing genetic adaptation to captivity is to limit the number of captive generations. So if you think about how you might do this, it's kind of pretty easy. If you're talking about plants, you can just store dormant seeds and you don't have to rear them, and you can let them wait, and you won't have this problem of many generations in captivity. However, in animals, this is more challenging. So one of the ways that breeding programs can try to do this is they try to breed older individuals so that there are fewer generations in captivity. So here's an example of the black-footed ferret, which is a species that is currently being managed in captivity. And if you see here, we think of this over time, and if you have an individual that's young, and then you mate them, and they're young and they get mated again, you have more generations in captivity than if you have an individual that you wait until they get older to mate them. You have fewer generations in captivity. So that's one thing that managers can do. But this can be difficult because in many animals, if they don't breed young, then they can have trouble breeding when they are older. Another way to minimize adaptation to captivity is to mimic wild habitats. So here's an example that's pretty interesting. In their wild habitat, elephants rely on the baobab tree, this large tree right here, for protecting against heat and offering shade. And the San Diego Zoo, in their new elephant enclosures, have mimicked that structure with non-living trees to provide similar functions so that they can maintain that memory of that natural resource. And finally, it's important to maintain equal mating opportunities. So this is important for several reasons, because when you have equal mating opportunities, you're mating all the individuals with all the other ones, and so you're maximizing genetic diversity. But also, if you think back to effective population size, if you are using equal mating opportunities, then you are making sure that each individual can contribute to that effective population. And so you're increasing the effective population size. So it's never easy, right? It can never be that easy. And we need to think about another interesting thing, and that is whether or not to manage small populations or large populations. So remember, small populations will increase the inbreeding depression, and that will reduce genetic diversity and potentially reduce reproductive fitness. Now, if we have large captive populations, the problem there is that's when there's an increased adaptation to captivity. So we actually want small populations to avoid this adaptation to captivity and large populations to avoid inbreeding depression. So what do? Well, the ideal is then a medium population size, and also to try to avoid inbreeding. And then another thing to do is to use a large population but split it up into smaller units. So here's an example of how that works. You have a large captive breeding population, and you split it up into several small populations, and then you basically wait and you let those small populations mate randomly until inbreeding becomes a problem, maybe an inbreeding coefficient of about 0.2. And at that point, what you do is you mix individuals from each of these small populations, and then again you wait and you allow things to go naturally until inbreeding again increases, and then you would mix it again, etc., etc. And then when the population is ready for reintroduction, you would then pool all those individuals for reintroduction, and you would actually have a higher effective population size. Now, another benefit of having these small populations is that it can avoid diseases. So if there is a disease that goes through, it doesn't wipe out the entire population, but maybe just influences one or two populations. And this sounds a lot like what zoos do. And so zoos can be quite useful for this, and they're kind of designed well to avoid adaptation to captivity. So we also need to talk about genetic management of reintroduction. So you've had a captive breeding population, you've increased the size of these, so how do you actually manage these reintroductions? Well, you have to think about several different issues. First one is you need to think about choosing the sites for reintroduction and the number of sites. And here it's important to think about, you want to match the original habitat where these populations came from. You don't really want to put them on the outskirts of the range. You want to make sure that they have the best chance of survival. Also, you need to think about what individuals are you going to choose for reintroduction and the number of individuals. So it makes sense to use healthy, young individuals that have lots of reproductive potential, and also ones that have lots of genetic variation and low inbreeding coefficients. Now, when you think about this, you also have to consider the fact that the individuals that you take out of that captive breeding program and put into reintroduction back into the wild, you will be losing from the captive breeding program. So you need to actually choose well which ones you're going to be moving. And here's an interesting graph that depicts this for the California condor. So all the dots are individuals, and along the x-axis we see here the captive population and the percent change in heterozygosity. So the positive, here we have 0, so the positive indicates that the percent change when individuals are removed from the captive population is positive, and the negative part means that the percent change in heterozygosity is negative. And here on the y-axis we have the reintroduced population. So here the positive means that the percent change in heterozygosity is positive when those individuals are added to the reintroduced population. And here these individuals, it's negative when those individuals are moved into the reintroduced population. And when you take a look at these quadrants, you have different ones, A, B, C, and D. They say different things. So for quadrant A, it is these individuals are detrimental when they're moved from the captive population, meaning that they are valuable to the captive population, but they are benefiting, they're positive in the reintroduced population, meaning they don't have a lot of similar alleles represented in that reintroduced population. For B, this is where those individuals the removal of them is not that bad in terms of the captive population because the heterozygosity would still be positive. And the addition of those individuals to the reintroduced population, it would be good because it's also positive, meaning that they would offer some new genetic material. So here in the C quadrant, removing these individuals from the captive population would be bad because it would reduce the heterozygosity, meaning that they're needed in the captive population for diversity, and introducing them into the reintroduced population is not really good because they're not really adding new or any beneficial heterozygosity. And then here in D, the quadrant, you have these individuals that removing them, it doesn't really matter. It doesn't really change the heterozygosity. It's still positive. That's because they have other individuals that have similar genotypes. But then it's negative for the reintroduced population because it is not really offering anything new. But when you think about initially introducing individuals into a population, you actually do want to end up choosing from D, this quadrant here. That's because you have really high mortality associated with releasing first releases of individuals into the wild. And so these individuals are the ones that can be most spared. They're not really adding anything to the captive population, and they're also they wouldn't really add anything to the reintroduced population. So if these individuals were lost, it wouldn't be such a problem. And then when time goes on and survival is better in the wild, then you could choose some of these more valuable animals that can be released into the wild to increase that population. After successful reintroduction, you still need to continue genetic management in the wild. You need to make sure that there's not inbreeding going on. Got to make sure that there's still genetic diversity and also gene flow between populations. And all of this is determined by having good pedigrees, so that is important. And then I should point out that most reintroductions are not successful. Only about 1/4 or 25% of reintroductions have been successful in the past. And we think of successful, we think of populations that have been moved into the wild and then can sustain themselves in the wild. And when we talk about management of reintroductions, we have to talk about hatcheries. So in theory, when we have hatcheries, it's to augment breeding populations in the wild. And this is usually considered to be a good thing. We've talked about this before, but there can be a problem with hatcheries. So it's called the Riemann-Laker effect, when you actually have increased abundance but decreased effective population size. And this decrease in effective population size is because in hatcheries, usually only 1 or 2 individuals are mated to produce many, many offspring. So you're reducing the number of individuals actually contributing to the offspring and to the effective population size. And then in hatcheries, they have lower reproductive fitness because of this adaptation to captivity. They've been raised in captivity, so they don't have the same selection pressures as the wild, and so they do poorly in the wild when reintroduced. And then there's also increased inbreeding depression because of the fewer number of individuals that actually did mate together. So there was more inbreeding. And it is possible to manage the genetics of a hatchery to try to avoid some of these problems. And this is where you would want that NE to NC ratio to be about 1. So you would want the effective population size to equal the census size. And the ways to do this, if we think back to a previous lecture, it is to maintain an equal sex ratio. You want to make sure you have random mating and you have equal mating between all individuals. And all these things will help contribute to each individual mating and producing offspring, and therefore being part of the contributing individuals to the effective population size.